this ideal situation, it would involve the joining of all of the exons or the retention of all of the exons and the removal of all of the interrupting sequences or the introns, followed by generation of the mRNA by the process known as transcription, which is followed by expression into a protein molecule or a polypeptide molecule, finally folding into the functional protein. So starting from this huge region, you're coming up with one protein based on how much of the region is actually getting focused on. Now imagine the number of genes that can be encoded from one chromosome and then think of the same from 23 different pairs of chromosomes. So information is immense, space is limited, so regulation is key. And a lot of that does indeed take place, some of which we have begun to understand, some of which is still beyond human understanding at this current point of time. But broadly speaking, these are the three types of regions that can be more commonly seen to regulate gene function, either a promoter, enhancer, or a boundary element is able to regulate the same. So if you look at this region for a promoter, you would see that there is transcription that can occur. Again, here if you see this is a mediator complex. Again, don't worry about any of this. I'm just giving you a perspective of how gene function proceeds. So there is a pre-initiation complex, there's a transcription factor and a mediator complex. All together, they form an assembly of proteins that helps in regulating gene expression. Technically, it can move in both directions, this transcriptional complex. However, it prefers or it preferentially moves more towards the 5' to 3' end, so that more and more transcription occurs from this region, whereas much less or almost no transcription occurs from the in the opposite direction. This is how normal expression occurs. However, when an enhancer is present, there is a difference whereby enhancer proteins help in recruiting more and more transcription factors. So if you see at the same position, more and more transcription factors get recruited, and that helps in controlling transcription. Then there are some boundary elements that distinguish different regions within the genome, and they are able to do so because they will block away certain regions and will bring certain regions close together, and they're able to do so because of cohesin proteins and CTCF, which helps in regulating this action. Therefore, it defines the start and end of a gene or start and end of a certain region within the chromosome and helps in differentiating um, specific regions of the genome at specific loci. So that was the organization of the genome. And sorry, can we just go to the previous slide? Yeah, that was the organization of the genome, the structure, the different elements that control it, and the different assemblies that can get affected by the proteins which are bound to the promoter, enhancer, or at the boundary element to delineate different parts of a gene. Now, this was the idea. These are the different ways in which gene expression can be studied. However, how gene expression is actually regulated is at all of these different steps. Commonly, 6 steps are known when eukaryotic gene expression is controlled. What are these steps? First, of course, you have the DNA, which undergoes transcription. So, transcriptional control is the first site at which a gene can be switched on or switched off. Following that, you have mRNA, which is formed from the transcript. So RNA processing control takes over. Okay, if you're not familiar with this, um, the default pathway for any gene which is undergoing transcription involves some additional steps, which includes polyadenylation and splicing. So splicing basically means removal of exons and the joining of introns, whereas polyadenylation involves the addition of a long poly A tail at the end of the transcript, which defines it or distinguishes it as a complete mature messenger mRNA, in contrast to the tRNA and rRNA and any other RNA which may be present within the cell. It gives the mRNA its identity and is retained. So any interruption in this step will be considered to be a disruption of RNA processing control, and that can also contribute to the regulation of gene function. Following this, from the nucleus to the cytosol, RNA transport and the localization of the RNA also makes a huge difference. Why? Because for translation, the next step, which is the production of a functional protein from the pre-existing mRNA, it requires the movement or the export of this of the mature mRNA from from inside of the cell towards the outside, and this majorly is regulated at two steps. Either there can be a disruption of RNA transport and localization within the cytosol, or there can be degradation of the mRNA. So even if successful transport has occurred, that mRNA might not be stable within the cytosol and might get chewed up by exonucleases, which will, of course, lead to loss of mRNA, loss of protein, and therefore gene expression will be hampered. Alternatively, translational control can also be executed at this step, whereby, despite the presence of an mRNA, translation may not be initiated at that site, or maybe preferentially less initiated at that site for that mRNA. So there also control can be executed. Um, if it is allowed to, however, proceed and final polypeptide is indeed produced, the activity of the protein can also be regulated by other proteins or by other factors. There can be certain repressors, there can be certain silencers, some which quench the activity, which remove the functional addition of those that remove the functional modification of those proteins. Examples majorly being in context of signaling pathways. Uh, if you have read about any of the cellular signaling pathways, you would know that phosphorylation, for example, is a major contributor to the functionality of many proteins within the cells, majorly the receptor tyrosine kinases. So the loss of of a phosphorylation on a particular protein can often lead to inactivation of that protein, and therefore protein activity can be controlled at that step. However, if all these steps can be bypassed and can be allowed to proceed without any interruption, then and only then is an active protein able to do its job. So gene function, gene expression, and gene function is very largely controlled at many different checkpoints within the cell based on the surrounding environment, based on the requirement and the availability of resources. So these are all of the different sub steps. So I hope it was clear. If not, you can let me know and I can help you out further. But yeah, apart from that, protein activity regulation is also regulated by covalent post-translational modifications that includes acetylation and ubiquitination as well. But of course, phosphorylation is what predominates in most circumstances, and it is quite abundant within the cell as well. So these are the different steps at which gene function, sorry, gene function can actually be regulated within the cell. Now, the gene control region for a typical eukaryotic gene. Now, this was, again, let's go back to the previous slide for some clarity. Yeah, so this was these were the steps at which gene expression is controlled. But if you come to the next slide, for a typical eukaryotic gene within the nucleotide sequence itself, there are several checkpoints that are kept in place before the expression of a gene is allowed to proceed. This is what we are looking at in this slide. So what are the different regions? First of all, for any gene, there is a promoter sequence. This, what you see here in light gray, is gene X, which is what we are studying. Sorry. So which is what we are studying, and right upstream of it is the promoter region. So the promoter is actually the DNA sequence where GTFs, or general transcription factors, and the enzyme which will catalyze this process, that is the polymerase. In this case, that will be RNA polymerase 3 because it carries RNA polymerase 2 because it carries out mRNA transcription within eukaryotic cells. So general transcription factors and the RNA pol 2, they assemble at the promoter site. The cis regulatory sequences are binding sites for transcriptional regulators. What do we mean by this? There are some cis regulatory sequences and there are some trans factors. What does it mean? Cis regulatory sequence is a sequence that is a stretch or is a portion of the nucleotide sequence that has regulatory function, and its function is executed by the binding of a protein which is binding in trans, which means it is not a part of the sequence itself but can bind to that sequence and control its activity. So that is a cis-regulatory sequence. They are very often sites for binding of transcription factors or transcriptional regulators. Sorry, whose presence whose presence on the DNA affects the rate of transcription initiation. These sequences can often be very close to the promoter, very far upstream of it, or even within introns or entirely downstream of the gene. So these regulatory elements, as you can see, are these light green sort of elements. They are present at various different locations, like the gene control region for GNX can be very close to the promoter, upstream, far upstream, or even downstream. The broken stretches of DNA, what you see here, they signify that the length of the DNA can vary, sometimes reaching tens of thousands of nucleotide pairs in length. So sorry. The Tata box is a DNA recognition sequence for the general transcription factor, TF2D, which is one of the initial proteins that bind and that initiate the process of transcription. DNA looping allows these transcriptional regulators that are present or that are bound at any of these positions to interact with the proteins that assemble at the promoter. So this is what you see here. This entire sequence on the left has undergone multiple types of folding to produce this architecture, whereby you can see that these far away sequences, all of them can interact with proteins at the center or at the promoter sequence. More often than not, it involves a mediator, which sort of acts as a scaffold for the binding of all of these proteins, but they're able to do this job. In some cases, they can also directly bind to the protein of interest by chromatin looping, but of course, that is circumstantial. That depends upon what type of condition, what type of protein you are studying, and it's very case specific. Okay, so many transcription regulators, they assemble or they act through the mediator, like I just mentioned, while some interact with GTFs and RNA polymerase directly. Several regulators also act by recruiting proteins that alter the chromatin architecture of the promoter. Whereas the mediator and GTFs are the same for all RNA poly2 transcribed genes, the regulator and the location of their binding sites will actually vary depending upon uh, depending upon which gene you are studying and which organism or which tissue you're studying it in. So the commonality between all of the RNA poly2 transcribed genes will be that the mediators are similar, the general transcription factors that are present for each gene would be similar. However, how they're arranged, where they are binding, and how they're interacting, that can vary depending upon what gene is being studied. But overall, the idea is that these are the multiple sites at which regulation of the DNA itself can occur, and how that is controlling the expression of the transcript of our interest, and whether or not the binding of all of these controlling proteins makes any difference to the final expression. Next comes yeah. Next come the six ways in which eukaryotic repressor proteins can actually operate. So what we saw till here was an overactivation or hyperactivation or increased expression of a gene. In contrast, there are a large number of cases where the expression of a gene needs to be controlled, retained, or completely silenced. So there are several ways. Majorly, here are the six ways. Again, you can refer to Bruce Alberts for further information about the same. But these are the six ways in which eukaryotic repressor proteins may operate within a cell. First of all, there can be competitive, or there can be competition between two types of proteins for binding to the same sequence. For example, here you see that upstream of the data box present on the promoter sequence of a particular gene, you can find a region that acts as a enhancer silencer site. Again, if you remember one of the previous slides, you saw, show it here. Yeah, the distal control elements, which are present either 5' or 3' of the gene of interest. You can find enhancer / silencer sequences, which indicates that they might possibly have competitive DNA binding at this site for enhancer and silencer proteins, and depending upon which is able to bind, which depends upon the abundance of the same within the cell and the kind of situation that the cell is in, the expression of a gene may be enhanced or may be silenced. So in this case, you can see that the binding site for the activator and the repressor actually overlap with each other, as a result of which there's a partial overlap. It's not completely overlapping, but there's a partial overlap, as a result of which if a repressor protein is able to bind, an activator protein, even though it is present within the cell, is not able to bind to the same site because it has already been occupied by a competitor protein, as a result of which gene function can be abolished. Not function, but gene expression can be stopped or can be hampered or declined under these circumstances. Second possibility is that the activation site can get masked. What do we mean by masked in this case? In this case, the binding site for the activator and repressor do not overlap with each other. They are segregated and quite far apart from each other. However, the two ends of the DNA binding protein are able to bind directly to each other, as a result of which the activating loop or the activation site is not able to bind to the mediator, which will help in completing the circuit that allows for gene expression to be enhanced. And instead, the repressor is quenching the reaction at that point of time because it is pulling away the activation surface and masking it from further use. So this is how activation surface can get masked. Third, yeah, DNA direct interaction with general transcription factors or GTFs can also occur. Here you have the data box onto which GTF-TF2D is bound directly, and because the repressor binding site is closer, when a loop is formed, the repressor binding site bound repressor protein is able to bind to the general transcription factor and prevents it from binding to the activator and prevents the direct interaction between these two or the activation of the gene of interest. So there is another level of control which is executed in this manner. So, yeah, that is occurring. And secondly, also, it is not allowing the assembly of other general transcription factors, which can allow the complete assembly of all the transcription factor proteins, and therefore gene function will be affected. Next, in panel D, sorry. In panel D, you can see that there can be the recruitment of chromatin remodeling complexes. Remodelers, again, not going into too much detail about it, but just so that we can have an introduction about it, which we'll discuss. Do not worry, we'll discuss all of this in much detail later on. But chromatin remodelers are able to bind to these sites. Again, see here the representation is in context of just the DNA because it is much more resolved. Whereas if you see here, this is for less resolved DNA where it is present in the uh, nucleosome format or the chromatosome format. And chromatin remodelers can come and remodel nucleosomes in such a manner that it can get, some regions can get sequestered. Recruitment of histone deacetylases can come in. Deacetylases, they, as the name suggests, remove the acetyl group from histones, and that helps or that inhibits or mitigates the expression of the bound DNA. Recruitment of histone methyl transferases. So HMTs basically they recruit the methyl molecule, which you can see in this green colored cap. These are proteins that bind to methylated histones, and as a result, in those regions, transcription cannot be activated, and therefore gene expression is significantly quenched. So overall, we see here that these are the common ways in which eukaryotic repressors may work. Either there can be competition between enhancer and silencer or repressor. Second, they can bind at their independent sites, but they interact with each other such that it is not able to, such that the enhancer is not able to do its job and its interaction surface is actually occupied. Thirdly, the repressor recruits a remodeling complex, which returns this nucleosomal state to the pre-transcriptional form, whereby other transcription factors cannot be recruited. Then the chromatin remodelers can come and bind, which prevent expression from occurring in that region. Repressor can also attract a histone deacetylase or HDAC to the promoter. Deacetylases remove acetyl groups, remove the negative charge present on histones, make it more positively charged, which allows DNA to bind a lot tighter via its backbone. Then a repressor can also attract histone methyltransferases, which recruit methylation gaps to which certain proteins can bind, which prevent its expression from proceeding. Additionally, these are some schematics again taken from Bruce Alberts for your reference. These are some schematics which show the properties of some other controlling regions, which includes insulators and barrier sequences. So what do they do? First, in this figure A, you can see that insulators, they can directionally block the action of certain cis regulatory sequences, whereas there are barrier sequences present here in green. Actually, both of them are green, but yeah, barrier sequences prevent the spread of heterochromatin. So what does this mean? Sorry. Yeah. So this is gene A, this is gene B. You can see that there are two insulator elements which are shown here in blue. There's a barrier sequence shown here in green, and this is some regulatory sequence which is controlling the expression of gene B. This is actively transcribed chromatin. What it can potentially do is that this insulator elements, they can come together, what you see in figure B. So insulators can come together when another protein which helps in bringing these two regions together, commonly called as an insulator binding protein or IBP, brings this chromatin or holds this chromatin in loops, favoring the correct cis regulatory sequence gene association. As a result, gene B gets properly regulated, and gene B's cis regulatory sequences are prevented from influencing the transcription of gene A. So I hope you understood this. Insulator is the job of an insulator is not to um, stop expression from happening, but to prevent the spread of the hyperactivation or the enhanced expression of a certain gene to the next one. So what it is doing is it is segregating out or removing that region which is going to actively undergo transcription, thereby preventing this accidental overexpression of another regulatory sequences, another gene by the same regulatory sequence, and by forming a separate loop. So that is what you see well represented in the second figure. In contrast, barrier sequences that I mentioned previously, they likely function as like they prevent heterochromatin from spreading forward. I've kept this figure 441 present in Alberts itself for your further reference if you want to know about it. So these are some other regulators of gene function that work physiologically under normal circumstances within a cell to control gene expression. In addition, there can be multiple inputs that are functioning at the promoter level as well. What do we mean by that? So, there are multiple sets of transcription regulators, co-activators, co-repressors, all together. What I'm trying to explain here is that it's not that there is one signaling pathway giving one stimulus or one go signal that can control the expression of a gene. There are multiple signals that a cell must sense, must integrate, and then finally make an informed decision about whether or not transcription should proceed. So here is what we understand, that when multiple inputs are present at a promoter, like for example, there's a neutral assembly of regulatory proteins. There can be a strong activating assembly. There can be a strong inhibitory assembly. There can be a weakly activating protein assembly. So all of that is taken into consideration. So yet, as per this book, when it was published, it was not very well understood how the cell achieves the integration of multiple inputs, but it is considered to be the result of a competition between activators and repressors. Whoever can bind more strongly, whichever is present in more abundance, can more often bind more strongly and instruct the cell to to proceed accordingly in that manner, thereby allowing the integration of all of these different inputs within the cell to finally regulate function. So, till now, what we have learned, this was the last slide from my end for today. I'm going to brush up on all the topics I've discussed for today. You can let me know if there are any doubts. I kept the last 20-25 minutes of the class for discussion, but since not a lot of people have joined, I think we can increase the material next class onwards. But overall, what we have discussed today, let's just take it again, back from the first slide. We talked about the discovery of DNA, the key experiments that contributed to the same, starting off with discovery by Frederick Miescher, where he discovered nuclein from leukocytes present in surgical bandages of patients. He concluded that there is some acidic portion which is called as which is why it's called as a nucleic acid, and there is some protein also which is affiliated to the same. Next came every MacLeod and McCarthy's experiment, where they injected the pathogenic bacteria, where they observed that non-pathogenic bacteria can be transformed by the genetic material of transformed of pathogenic bacteria. So disease-causing bacteria obviously causes disease. Non-disease causing bacteria, which previously did not cause disease, can also do so when transformed with the DNA of pathogenic bacteria. So they basically concluded that it is DNA which gives the ability uh, to these bacteria to cause disease and therefore execute the inheritance of information from another system. This was confirmed also by Frederick Griffith, and this was further confirmed by Avery and his colleagues by the completion of this experiment. Additionally, Hershey-Chase experiment showed that it was indeed radioactive material. The radioactivity of a particular virus is carried forward into the cells via via the genetic material and not via proteins. And the genetic material in this case was DNA because it was P32, which is the phosphorus that gets that gets inherited across species from the virus into these cells, and therefore it is actually DNA which is carrying this genetic information, whereas the protein coat is simply there for structural stability and for formation of a syringe-like structure, which allows bacteriophage to inject its DNA. So Hershey-Chase experiment was further confirmation of what was already being studied at that time. Then the structural organization or the architecture of the DNA molecule was understood in extensive detail based on X-ray diffraction experiments by Rosalind Franklin. And the interpretation was carried out by Francis Crick and James Watson, who were who, along with Maurice Wilkins, were awarded the Nobel Prize in Medicine, and they were able to decode Rosalind's data, thereby giving them thereby getting them their Nobel Prize. You can read more about this. There's a book actually called "The Story of Photo 51," if I'm not wrong, which will give you a lot more perspective about it. And also, that is how the structure of DNA came to be known. Then more details about the DNA structure, building blocks, which are nucleotides, how they are arranged, how the sugar phosphate backbone gives it the stability, whereas nucleotide bases, they project towards the inside. They project towards the inside, and they are hydrogen bonded to each other based on purines and pyrimidines, which are aligned along the sugar phosphate backbone. And this hybridization is what gives the DNA molecule its extensive stability. This hybridization property was explained a lot better by Chargaff's rules, where he concluded that that AT and GC are actually hydrogen bonded. Therefore, the effective number of adenosine and guanosine molecules is equal to the number of thymine and cytosine molecules. And that actually showed how complementarity is so crucial towards the formation of the final structure of DNA within a cell. And also, other rules by Chargaff included the fact that base composition does not change with the age, nutrition, or the environment of a living cell. However, it can vary from one species to another, but is considered to be similar within different tissues of the same species. So those were Chargaff's rules. Then came Watson-Crick's model for DNA structure, whereby they showed that there are two helical chains wound around each other to form a right-handed helix. The hydrophilic backbones consist of deoxyribose sugars and phosphate groups, whereas towards the center are the purine and pyrimidine bases that are stacked one on top of the other. And the pairing of these two strands creates a major groove and a minor groove within the DNA in the helical duplex. Then we understood the key definition of a gene, how it occupies a position on the chromosome. We understood the organization of the DNA within the cell, how its compaction is carried out from the 2 nanometer wide DNA double helix through the formation of a nucleosome by interaction with histone octamers around which DNA binds 1.865 times, following which there is a stopper like H1 histone, which forms the chromatosome. The chromatosome folds up to produce a 30 nanometer fiber, and 30 nanometer fiber is able to form loops that average around 300 nanometers in length. Finally, they are compressed to form the arm of a chromatid, arm or the chromatid of a chromosome, which is what we visualize within the cell. And also within the cell, most of the information that you find within the chromosome is actually considered to be non-protein coding, but not redundant. They are all involved in regulation in many different manners. A significant portion is contributed by introns, whereas the largest portion is contributed by transposons, different transposable elements like retrotransposons, DNA transposons, signs, lines, etc. And a very small percentage, around 1.5%, includes these protein coding genes. Then we looked at these chromosomes. This is a GEMSA stain for the chromosomes, the complete set of human chromosomes, where we see different sizes of each of them, not just size, but morphology of the same as well. And how it is relevant is that we understand this in under default circumstances is how chromosomes should ideally look. But if there are individuals that carry some sort of chromosomal aberration, then you will be able to see that there is a change in the staining. For example, if there is reciprocal translocation, I don't have an image for it, but it has been extensively studied and has been reported previously as well, whereby if you see that one portion of a chromosome has fused to some other portion, you'll be able to colorimetrically distinguish it because, uh, say, for example, chromosome 17 and chromosome 2 have reciprocal translocation, then a part of this red chromosome will be present in the blue, and the blue part gets exchanged with red. So such staining helps us to identify chromosomal translocations that might have occurred, and that gives us an idea about how the genome of that individual looks. Then we discussed the central dogma of molecular biology, whereby we understood that DNA to mRNA to protein, which was the default hypothesis, has undergone a lot of change over time. DNA replication has been included, reverse transcription has been included, and prions are also considered to be an exception to this. There are several other small changes to it as well, but majorly when we talk about the dogma, this is what the dogma looks like. Uh, then we looked at the gene structure, whereby we understood that the actual coded region or the actually transcripted and translated region is very small. Not transcripted, transcribed region and translated region is very small, whereas the regulatory sequences downstream and upstream of it constitute the major part. There are some proximal control elements very close to the promoter, some distal elements very far from it, either at the 5' or 3' end. And both of these are able to regulate or control the expression of a certain gene. Um, when we looked at the chromosomes at large, we saw that for entire chromosome is actually coding for a particular gene, where exemplarily we took chromosome 22, and we see how 10% of the arm only has 40 genes and 1% of the arm only contains 4 genes. Within that one gene, it has around 3.4 into 10 to the power 4 nucleotide pairs, most of which are also exons that get introns that get removed. Exons, they join up and they encode for the RNA that produces protein and finally undergoes folding to execute its function. So a huge recap of all that happens within the cell. Then there are different types of elements. The ways in which these different types of elements function also include promoter activity, which help in normal transcriptional function, enhancers, which recruit more number of transcription factors. Here you can see around the mediator complex, thereby controlling transcription at these sites, and boundary elements which help in looping out certain regions of the chromatin and segregating or creating boundaries within the entire DNA to preferentially favor the transcription of a certain region. An example of which we saw later on, where insulator elements were also assisting in regulating gene function in this manner. Um, then we looked at the different steps at which eukaryotic gene expression was controlled, either at transcription, RNA processing, transport, and localization from the nucleus into the cytosol, control at the level of translation, regulating mRNA degradation, and finally protein activity control. When all of these are bypassed and finally active protein is synthesized, then that allows the gene function to properly proceed. So there are a number of post-translational modifications also at this step, which include phosphorylation, acetylation, or ubiquitination, all of which can help in controlling the expression of a functionally coded gene. And then we looked at the then we looked at the gene control region, the different elements which contribute to the same. We looked at some co-activators and transcriptional regulators, which can either bind upstream or downstream of the sequence, and how when we look at the 3D conformation of this regulatory region, we can see that there are some possibilities whereby these far off distal elements can also interact with the promoter to preferentially control the expression of a gene by means of a mediator protein, which brings them close together, or by indirectly brings them close together, or by direct interaction with the promoter sequence as well with the help of co-activators. So that is another possibility by which gene expression can also be controlled. Um, we also looked at eukaryotic repressor proteins and their mode of action. So they can competitively bind to the same DNA sequence whereby their binding sites are the same for the activator and for the repressor, or there can be masking of the interface via which these two proteins can interact, thereby repressing gene expression. There can be direct interaction of the repressor protein with general transcription factors like TF2D. There can be recruitment of some chromatin remodelers which wrap up these sites and prevent transcription from proceeding. There can be histone deacetylation, which can be carried out by HDACs or deacetylases, which prevent or which uh, wind up the DNA further and prevent its expression from taking place. Or there can be histone methyltransferases that recruit or that methylate certain regions of the proteins that bind to these methylated histones and that prevent transcription from proceeding in those sites. So these are also different eukaryotic transcriptional repression methods. Finally, we understood some other regulation carried out by insulators and barriers. So insulator elements present across a gene help in looping it out from the rest of the chromatin, bringing the two insulator binding sites closer and preferentially favoring the expression of a certain gene over the other to prevent any cross transcription among different genes. Barrier sequences, on the other hand, are expected to prevent the spread of heterochromatization any further. So whether or not that region will undergo active transcription or not will can also be dictated to a large extent by these barrier sequences. Finally, we discussed the possibility that transcriptional initiation is not the outcome of only one type of signal, but instead multiple inputs that undergo integration at their site, whereby some favor strong activation, whereas some favor inhibition, some some undergo modulation based on the binding of other trans factors to these sites. And it is the comprehensive integration of all of these signals that altogether the cell decides whether a gene should be expressed or not under a certain circumstances within the physiological state that it is in. So with this, I will conclude my lecture for today. If there are any doubts, please let me know. We'll discuss them and then end the class for today. If you have any questions, you can share them, or if you have any inputs, anything you would like to be done differently. If you want some questions, some takeaway questions, or something like that, we can do that as well, and that can be implemented from the next class onwards. I'll uh, try to uh, I'll try to include a little more information since we have some spare time on hand from the next class. But for now, we can maybe keep it at just to this much. We'll begin a little bit by methylation of DNA, and because that is not something I discussed a lot, I talked about histone modifications, but not about DNA methylation. So I'll mention that. And then we'll move forward with mutations, what role they have in it, how they're different from polymorphisms, and how they can actually be studied as a part of functional genomics and what relevance it actually carries. That we can discuss from the next class onwards. So thank you so much for joining. You can mail me if you have any if I can help you out in any way, if you need any information, any study material, or any help for that matter. And lectures will be uploaded on YouTube in a couple of days, and I'll share the link for that as well. Please try to join live lectures and please try to interact. Don't hesitate. We are all students, we're all learning, and we can have a good discussion next class onwards. Please also note that next class will be on Tuesday, which is 28th of October. Uh, if in any case, a lot of people, the registered participants, if most of them have any suggestion about a different day for hosting classes, you can let me know. I can schedule it for Wednesday or any other day as per everyone's convenience. So, with this, I'll end today's class. Thank you so much for joining and keep studying. Thank you and good night.
Thank you, ma'am.
Thank you, ma'am.
Bye. Okay, bye.